but oh well, let's go. Number 6, true or false? People with heterozygous sickle cell trait are resistant to malaria. This is very true, right? So we should understand the fact that a sickle-celled anemia, right? So a sickle-celled red blood cell, because we're talking about red blood cells here. Oh, I see a question. Yeah, you can speak, Kimberly. Yes, Kimberly. I see a hand. Oh, it's a mistake. Is it a hand? Is it a hand, Kimberly, or we should move on? I think we should move on because I don't see you responding. All right. I was talking about what? Question 6. Is it clear? Yeah, I think it's a. Let me try to zoom in a bit more. All right. Yeah. All right. So we're talking about number 6, right? So we're saying people with heterozygous sickle cell trait are resistant to malaria. I said this one is true. Why? When you're talking about sickle cell, here, since it's blood physiology, we're talking about blood, right? And the type of blood cell we are talking about is red blood cells. So red blood cells, right, they can sickle if they have a poor oxygen supply. Right, they can get a sickled shape or a shape of a sickle, right, which is kind of this bent knife thing, right? So when one has a heterozygous sickle cell trait, right, it means that when the mosquito bites them, right, and infuses the plasmodium of malaria, it grows in what? In red blood cells, and causes them to sickle because it kind of destroys the hemoglobin. When it destroys the hemoglobin, it causes the sickling of the what? Red blood cells. So when one has the sickle cell trait now, it means although having the sickling is not severe, right? And if there is a sickling now, the plasmodium cannot live in that sickle celled what? Red blood cell. If they cannot live in the sickle cell red blood cell, it means one cannot have what? Malaria. And the malaria plasmodium cannot what? Spread. So in summary, we are saying one who has a sickle celled red blood cell cannot have malaria. Why? Because the pathogen or the disease causing organism which causes malaria cannot grow in that sickle celled what? Sickle celled red blood cell, right? Then B, folic acid is not required for erythrocyte synthesis. This is false. Folic acid along with vitamin B12 are necessary in the production of red blood cells, right? Then C, serum is plasma with no clotting factors. Although this might sound true, it's actually false. Serum is plasma with no specific type of clotting factors such as fibrinogen and the likes. There are 4. But it's specific type of clotting factors, not all clotting factors, right? Then D, pale skin can be a symptom of anemia. This is true. We know that red blood cells, they have a red color, right, when they are in blood vessels. So whenever they are few or their concentration of hemoglobin is few, one has a pale skin, especially in those who are light skinned. But those, sorry, what was the answer for C? C is actually false, because we are saying that serum is plasma with, some texts say plasma without fibrinogen, but it's actually plasma without certain types of clotting factors, which are 4, right? We can go and search the other ones. There is fibrinogen 1 and other 3. Okay, then D. Okay, that's okay. Then E now. Estimated sedimentation rate is increased during inflammation. This is true, right? Because when you're saying sedimentation, we're just saying that we take our blood, right, then we centrifuge it. When I say centrifuge, I mean we separate it into components from white blood cells, red blood cells to plasma. So when one is in a diseased state, right, they tend to, when we centrifuge, you see that the red blood cells, they tend to sediment down fast, and the white blood cells, the puffy area, the puffy coat, tends to ascend up high at a quicker rate because of what? The inflammation. Whenever there is inflammation, we are saying that the immune system is very, very active. So are there any questions on number 6, or we should move on? Yes, I see a question here. Number 3A. Number 3A. Yes. Oh, number 3A. Is it number 3A or? 3A. Yes, the following are major hematopoietic growth factors. Is that the one? Yes. Yes, what I can't. But that's the one that's intrinsic factors, not in hematopoietic growth factors. Sorry, can you come again? Yes. Okay. Intrinsic factor, right? It does not contribute to hematopoietic growth. It's not a hematopoietic growth factor. It is actually a transporter. It transports vitamin B12 from the stomach to the ileum, and from the ileum is then reabsorbed and sent to the bone marrow. So it's a transporter. It's a transporter. It's not a growth factor. All right. Sharp. What is the answer for 6E? All right, let me go to 6E. 6E. 6E is true. I say that, let me just show you the slide, this one, which talks about sedimentation. All right. Yeah. So this is like the centrifuging of blood, right? So when you centrifuge blood, we see that there's red blood cells, there's a puffy layer of white blood cells, and there's a plasma which is above. So when one is in a disease state, we see that the sedimentation of red blood cells is very fast. It goes down very fast, right? And we see that the white blood, like the puffy coat which contains white blood cells, is very prominent. So this is why we said in a disease state. So in a disease state, your immune system is very active. So you expect this part to be very prominent, and the rate at which red blood cells go down is very fast, to allow the immune system to kick in fast. So now let's move. We're now on number 7. The following are clotting factors, right? So clotting factors are factors which are involved in the coagulation of blood. So when you say coagulation of blood, we're just saying we're trying to stop blood from leaking, right? That is the essence of hemostasis. So A, fibrinogen, this is true, which is clotting factor 2. Then prothrombin, this is again true, right? It's the precursor of thrombin, right? Which is a clotting factor. Then tissue factor, again, this is true, this is clotting factor 3, right? Then calcium, true, right? It's the clotting factor 4, right? All these are clotting factors except thrombomodulin. Thrombomodulin is not a clotting factor. it is actually involved in the breaking down of clots, right? It's a transmembranal protein which cleaves a certain protein, right, which helps us in the breakdown of what, of the clots. It makes blood move. It's a anticoagulant. So was that clear on number 7, or should we move on? Okay, let me admit people here. Any questions? Is there any question on number 7? Yeah, so I think let's move. Number 7 seems to be clear. Alright, Mudei, I saw a question. Is it a question? Is it a question, or should we move on? Yeah, let's move. I think everything is clear. Alright, the following are factors of the intrinsic pathway. So the intrinsic pathway, right, we have two pathways, right? We have an extrinsic pathway and an intrinsic pathway. The extrinsic pathway is shorter and the intrinsic pathway is what? Is longer. And some texts believe that the extrinsic pathway was derived from the what? From the intrinsic pathway, right? Because we see that the intrinsic pathway is major what? Major, it has most of the clotting factors, and the extrinsic pathway happens as a result of external forces, and the intrinsic pathway just happens internally, right? Then the clotting factors, right, which are involved in the intrinsic pathway. Let me just show you this table so that you can just appreciate. Let me show you this one. Yeah. So the intrinsic pathway and the extrinsic pathway. Can you see that the intrinsic pathway involves clotting factor 12, 9, 12, 11, 9, and factor 8, right? Then they all converge to a common pathway, which is factor 10. Then the extrinsic pathway involves, like, you'll be cutting now external source, right? Then the intrinsic pathway is just an internal cascade. Extrinsic pathway is due to the cutting of the what? Of the blood cell. It activates tissue thromboplastin, right, and tissue phospholipids, right, which results in a clotting factor, which is tissue factor, or factor 3, which activates factor 7 to its activated form, and this factor 7 forms the common factor, which is factor what? 10, which helps us to form a fibrin, what? Eventually a fibrin meshwork. So we are seeing in simple terms the intrinsic factor is factor 12, factor 11, factor 9, and factor 8. Those 4. Now we go back to the question. The following are factors of the intrinsic pathway. Tissue factor, false. We saw it in the what? On the extrinsic pathway. Then factor 7. This again is false. We saw it on the what? Extrinsic pathway, not the intrinsic pathway. Then factor 12, that was true. We saw it in the intrinsic pathway, which we said is longer, right? So it should involve all bigger numbers. Then D factor 9, this is true. We saw it again. Then E factor 8, we saw it again. The reason why we do these pathways, just for you to appreciate, we are saying that the first thing which happens whenever you get a cut, right, there are some mechanisms which just happen normally, right, which involve your platelets going to stick on the what? On the cut, so that there is no blood flow from your blood vessels to the external environment, thereby maintaining hemostasis. But you should take note that these platelet aggregation, right, is not enough. That's why we use we use clotting factors. Clotting factors act like a reinforcement to the platelets, because the platelets are not enough on their own. That's why we have the extrinsic pathway and the intrinsic pathway. The intrinsic pathway being the major pathway, right? Are there any questions on the intrinsic pathway before I said number 8? Yeah, we're on number 8. We're talking about the coagulation cascade, right, which happens intrinsically and extrinsically. Extrinsically due to what? External forces. For example, cutting. I think did you see this image which I just used? This one. Extrinsic pathway involving cutting. Can you see? We are cutting the cells, right, which are called we're cutting the cells which are near the blood vessel. So these cells they release calcium, and this calcium is necessary for what? For the formation of this tissue factor, right? Then the tissue factor then activates factor 7. Then the activated factor 7 forms our common pathway, which is the factor 10. The factor 10 now forms a complex which converts prothrombin, right, or the inactive form of thrombin, into thrombin. Then this thrombin now converts fibrinogen, right, into fibrin. Then the fibrin now is a meshwork. It's like, I wish I had a better diagram. It's a meshwork which kind of is something like this. It's this meshwork, right, which kind of glues together the platelets so that they remain in position. Something like that. I think it's a bit clear now. I think it's a bit clear. Do I have a better diagram? Yeah, this one, this threadish fibrin meshwork, this is the result of the what? Of the coagulation cascade, right? It forms part of the secondary hemostatic mechanism. The primary hemostatic mechanism just involves your platelets aggregating here and stopping blood flow from blood vessels. But now the secondary now hemostasis, where we see that things now are not okay, we need a reinforcement, we use the clotting factors. So yeah, now let's move. Are we now on the same page? So we can move, right? Alright, no. Are there any questions? Let me see. What about factor 5? Is it a cofactor? Alright, okay, let's just look at this. Look at this. As you can see here, can you see any clotting factor 5? Clotting factor 5, yes, its main importance, right, is to help us to form this complex. This complex is the one which forms the enzyme, which is the prothrombinase complex. The enzyme now which helps us to convert prothrombin to thrombin. Thrombin now helps us to form the fibrous meshwork, this one, right? So factor 5 is a factor, yeah, it's a clotting factor, right? But it's not in the intrinsic pathway or the extrinsic pathway, but it helps us to form this intermediate compound, right, which helps us to catalyze the formation of a fibrin meshwork. I hope it's clear, Chan. Yeah, I hope I answered you, Rutendo. Also the answers, sorry, I forgot the answers. So A is false, B is false, C is true, D is true, E is true. So let's move to question 9 now. We have to finish maybe maximally by 10, because we're supposed to finish by half 9. Right, the following factors oppose clot formation. So the factors which oppose clot formation, I see a hand. Okay, Michelle, you can unmute. I see a hand. Yeah, you can unmute. It's okay. You can speak, Michelle. I see the hand. Hello, are you still available, or you should? Let's move on, boss. Yeah, yes, you can speak. You can speak. I saw the hand. Yeah, you can speak. Yes, you can unmute. You can unmute. It's okay. You can unmute and speak. Yeah, pane timu rakati nichangoribatsira but ndikutori. They break down the fiber meshwork. So the following are factors that oppose clot formation, right? Let me just show you the let me not say the answers, it won't make sense. So yeah, now it's called, the process is called fibrinolysis, right? So when I'm saying fibrinolysis, we're saying we're just breaking down the fibrin meshwork, right? We are condensing it. Let's say, for example, the clot has served its purpose. So when the clot has served its purpose, we have to remove it, right? So what do we use? We use the modulator, which I talked about earlier, called thrombomodulin, right? So this thrombomodulin, it helps us, right? So thrombin is a clotting factor, right? Thrombin, the clotting factor, we take it now, then we inactivate it. We take it, we inactivate it. We inactivate it using what? We inactivate it using thrombomodulin, protein C, and protein S. So these now are called anticoagulants. They prevent the coagulation cascade or the formation of a fibrin meshwork, right? They prevent the formation of a clot, right? So in summary, we have thrombomodulin here, right? Protein C here, then the protein C and the protein S, right? They form a complex which inactivate factor 5 and factor 8, and protein C inactivates factor thrombin. It inactivates thrombin, right? Again, this is another diagram. We have protein C, right? Protein C now, it inactivates factor 8, which is said is part of the intrinsic pathway. Factor 5, which helps us to I think someone is entering. May you kindly share the slides? It's okay, I'll share the slides. But the things which are in the slides, we explained them in the videos. There are some videos which we sent. They have all of this in them, so try to check them out. They are on there, they are on YouTube. As bro ano chozokumbira this book, it's okay. Oh, the network, I'm recording this session, so I'll just send it through on YouTube. All right, so now let's go back to the question. We have to move. I have to move, right? So can you see here, this is the pathway which results in the breaking down of the fibrin meshwork. So the fibrin meshwork, right, is broken down by breaking down the clotting factors. So now protein C, now the activated form of protein C, right? It breaks down factor 8, which is part of the intrinsic pathway. It breaks down or inactivates factor 5, which is very essential in the formation of that enzyme which I said converts prothrombin into what? Thrombin, right? It also inactivates uh, inhibitor of tissue plasminogen activate. So when it inactivates the inhibitor of tissue plasminogen activator, we then convert plasminogen, which is inactive form, into plasmin. Plasmin now helps us to break the fibrin meshwork. This fibrin meshwork, right, is broken down by plasmin. Plasmin helps us to break down the what? The fibrin meshwork. So this one is a simpler diagram, but I'll just send the slides so that you can just understand. Right, the plasmin is helping us to break down or dissolve the clot into just fibrin degrades. So now we go back to the question. The following are factors that oppose clot formation. Protein C, protein C, that's true, right? Factor 7, it's false. Factor 7, we saw it in the extrinsic pathway. Then C, tissue factor pathway inhibitor, right? Tissue pathway factor pathway inhibitor is true, right? Can you see it here? Tissue factor pathway inhibitor. No, it's not here, but yeah, let me look for another diagram. Yeah, this one. Tissue factor pathway inhibitor, this one. It inhibits the extrinsic pathway, so it opposes the formation of a clot. Tissue pathway factor inhibitor is produced and it inhibits factor 5, factor 7, and this complex inhibits coagulation. I think, yeah, that's clear. Then what else? Thrombomodulin, right? Again, thrombomodulin is true. I showed you thrombomodulin. Then antithrombin 3 again is true. I'll just send the slide so that it makes sense. So let's move to number 10. The following are anti-clotting drugs. Anti-clotting drugs. So anti-clotting drugs, they prevent formation of what? A clot. We have aspirin is true. Let me see. Someone wants to. Alright, so we have aspirin, right, which is true. We have recombinant TPA. This is the recombinant TPA. Let me show you from my slide. Where are you? Recombinant TPA, which we said is part of where are you? Here. This one, the TPA, this one, right? The TPA, this one. Can you see it? It helps us to break down the fibrin meshwork. So it's a type of what? Of anticoagulant. So when it's recombinant now, it means it's not naturally found, but we are now taking it from the pharmacies and stuff. You guys, the pharmacists, you form these recombinant TPAs. Then streptokinase, again, that one is true. It's explained in the slide, guys. Don't worry. Just know that it's true for now. Then DSPA, that one is also true, right? Can you see there is plasminogen? So this compound helps us to form plasminogen. Plasminogen is necessary for the formation of plasmin, which is said plasmin helps to break down the fibrin what? Meshwork. But you just have to appreciate these things. Just appreciate that there are things which are anti-clotting. The major examples which you know by heart is aspirin and heparin. These other three, the lecture will just be if, but just know these major two. They are the major ones. So everything on number 10 is true.